Hello everyone. Welcome back to Inside Chlorpromazine, where we explore the unexpected intersections of this decades-old drug with modern medical challenges like COVID-19 and long COVID. Today's episode is titled Viruses and Vesicles, How Chlorpromazine Blocks Viral Entry. And I must say, this might be one of the most fascinating mechanisms we have covered so far. Joining me again is Chaitanya, a microbiology researcher whose work focuses on cellular infection mechanisms. Welcome back to the show.
Thanks, Sowmya. I have been looking forward to this one. The way chlorpromazine disrupts viral entry in such a complicated, compelling part of the story.
Let's start at the beginning. How do viruses like SARS-CoV-2 actually enter our cells?
Viruses are masters of deception. They attach to surface receptors on host cells. For SARS-CoV-2, the ACE2 receptor and trick the immune cells into engulfing them. This process usually involves a cellular mechanism called clathrin-mediated endocytosis. Think of it like the cell forming a double bubble or vesicle around the virus and pulling it inside.
So, Catherine is kind of like the scaffold or machinery.
Exactly. Clathrin proteins form a lattice structure on the inside of the cell membrane that helps shape that vesicle. Without clathrin, this whole entry mediate method breaks down.
So, where does chlorpromazine come in?
Chlorpromazine is known to inhibit clathrin-mediated endocytosis. It interferes with the assembly of clathrin-coated pits by redistributing clathrin and adapter proteins away from the plasma membrane. This means that viruses that depend on this mechanism, including coronaviruses, are unable to successfully enter cells. Essentially, chlorpromazine keeps the front door locked.
Has this been observed directly in studies?
Yes, several in vitro studies have shown that chlorpromazine can inhibit the entry of SARS-CoV-2 into human cells. It's not just theoretical, it actually blocks infection in cell cultures. And what's more, this isn't limited to SARS-CoV-2. Chlorpromazine has shown broad-spectrum antiviral effects against other viruses like MERS-CoV-2, Ebola, influenza, and even HIV, all of which use similar endocytotic routes.
This seems huge, stopping the virus before it even gets into the cell.
Absolutely, that's the ideal strategy. If a drug can prevent viral entry, it may reduce viral replication, symptom severity, and potentially even transmission. Of course, this has to be balanced with drug concentration, timing, and safety in real life human tissues, which is why clinical studies are essential. But conceptually, it's very powerful approach.
Could this mechanism have relevance in long COVID?
That's fascinating and evolving area. One theory for long COVID is persistent, low-level viral reservoirs in tissues. If chlorpromazine can block viral entry or re-entry into susceptible cells, it may help in clearing or controlling these residual viral particles. Additionally, its anti-inflammatory properties could help manage the chronic symptoms driven by immune dysregulation.
Are there limitations to this antiviral mechanism?
Yes, of course. Chlorpromazine activity is primarily relative. It's most effective when used at or before viral entry. Once the virus is fully inside the replicating, other drugs or immune responses are more relevant. It also doesn't affect all entry pathways. Some viruses can bypass clathrin-mediated endocytosis using caveolae or direct fusion. So, chlorpromazine wouldn't block those as effectively. And again, we are talking about cell culture data. not large scale human trials, so more evidence is needed.
It's still impressive that such an old drug can have such modern applications.
It really is. Chlorpromazine is a great example of drug repurposing, taking a well characterized medication and giving it in a new life in a different field. It's also a reminder that we should never underestimate the complexity of existing drugs. There is still so much to learn.
To wrap up, chlorpromazine's ability to block clathrin-mediated endocytosis makes it a strong candidate for preventing viral entry, a crucial step in the infection process.
Exactly. It doesn't just treatment symptoms, it potentially stops the infection at the gate.
Thank you, Chaitanya, for that clear and detailed explanation. This episode definitely adds a new layer to our understanding of chlorpromazine in COVID-19.
Always happy to join Sowmya.
And thank you to our listeners for tuning into Inside Chlorpromazine. Don't miss our next episode, immunomodulation or immunosuppression, chlorpromazine's role in the cytokine immune conversion. We'll explore whether this drug calms or confuses the immune response during infection. Until next time, stay informed, stay safe. Thank you.